through the project I'm going to talk to you about. The first thing I wanted to do is actually to introduce the Midlands Midlands Mass Spectrometry group to you. So just to say something a little bit about where that's come from and what it is. I'm sure all of you will be familiar with Midlands Innovation. Midlands Innovation is a collaboration between a number of universities across the Midlands to promote research and to strengthen the research base, and to do other good things like in strengthening our technical support. About over a year ago now, we started, or a couple of people started thinking about putting together the Midlands Mass Spectrometry group. And the idea behind the Midlands Mass Spectrometry group was actually to bring together researchers and facility managers and people who use mass spectrometers across the Midlands to engage with that community, to engage with the user community, to provide a knowledge base so that the people who were interested in using mass spectrometry could come to that knowledge base of experts, but also provide an access route. So if people needed access to particular techniques or technologies across the Midlands, the mass spectrometry group would be an opportunity for people to access that. Sharing best practice was one of the key areas, so making sure that we were doing things as well as we can. But perhaps one of the real strengths of it is to provide a forum for training and knowledge transfer, and this is especially for technical staff. So we're very keen to make sure that we have the technical staff learning across the Midlands from each other and forming a strong technical staff base to support research. One of the good things that has come out of the Midlands Mass Spectrometry group is that it's become a platform for collaboration rather than competition. So this is all the universities working together to build the Midlands Mass Spectrometry capability, and this means that we've been working together in making applications to funders to make sure that we get the maximum impact for the money that the funders are putting in to support mass spectrometry in the Midlands. And this has been very successful in bringing some of the latest instrumentation to the Midlands. But it's not just the Midlands that we work with, so we're working with the wider mass spectrometry community across the UK, of course, and across beyond that, so across Europe and the world, and to promote the use of mass spectrometry in research. And I must say, this is not just biological mass spectrometry, this is all aspects from petroleum right the way through to drugs and small molecules. So anything that uses mass spectrometers, we have expertise across the Midlands in all of those areas. But what we're doing today is this bottom point. We also wanted to provide a forum for discussion of mass spectrometry methods and its use in research so that people could access that. And that's the point of today's seminar and the seminar series that we've just started, so this is the first of that seminar series, is to have some scientific talks, but also some technical talks and some information talks to let people know what's going on in mass spectrometry research across the Midlands. The group does contain a number of universities, they're represented here on this slide, and a number of people from those universities. I should mention specifically Alex Jones and Andrew Bottrell from Warwick, who were instrumental in setting up the mass spectrometry group. Liam Heaney, who took on chairing the mass spectrometry group. I'm speaking to you today as the acting chair of the group, while we seek a more permanent chair to take over from Liam, who's finished his time as chair. If you want to learn a little bit more, then there's a link at the bottom of this screen that you can go to, and that will take you to the Midlands Innovation page where the mass spectrometry group is hosted. There's also information there about things like the mass spectrometers that we have available across the Midlands. So that's what I wanted to say about the Midlands Mass Spectrometry group. If you're interested in what's going on, then do look at the webpage, but if you want to get any more information, then do feel free to contact either your local representative or to contact me. And if you're interested in getting involved in any way, then please do contact me, and I'll see what we can do. Okay, so that's the Midlands Mass Spectrometry group, which is the leaders for this seminar. What I wanted to do next is just to take you through where some of the work that we're going to talk about today came from. And this came from the NHS Test and Trace UK Moonshot programme. Some of you may remember in September last year, Boris Johnson, who is clearly learning about mass spectrometry there, looking at how excited he is by this, announced the Operation Moonshot, which was a $100 million programme to try to boost the UK's capacity for diagnosing COVID. That very quickly got subsumed into the NHS test and trace system. And so as part of that, we applied to, or Perdita Barron led an application to the Moonshot to use mass spectrometry as a diagnostic for COVID. If you're wondering why they called it a moonshot, then I think the picture on the right here explains it. The first is that moonshot means the goal is a very, very long way away. And the second is that all those craters are the shots that missed. And there were plenty of those along the way in this project. But what we're going to tell you a little bit about today is what we've been doing as part of this moonshot project and some of the associated COVID research that's been done in the Midlands associated with that. And also tell you some of warts and all about what didn't work for some of it. What this came out from originally was at the start of the COVID outbreak, Perdita Barron led a coalition of mass spectrometrists across the world called the COVID-19 Mass Spectrometry Coalition to see how we could use mass spectrometry to benefit research in SARS-CoV-2 and benefit the community from that. And this started off as a worldwide collaboration. There are now over 400 members of this coalition, and you can see in the map down on the left-hand side the density of those. Most are in the UK, but you can see there is coverage across the world effectively of people who are involved in that coalition to use mass spectrometry for COVID research. Why did we think that mass spectrometry would be good for this? Well, why another test for SARS-CoV-2? Well, partly it's cost. So the PCR test is relatively expensive, about £50 to £90, depending on who you talk to. But we're also looking at whether we could have speed, capacity, and also avoid some of the supply chain problems that the original PCR test had. So the challenges we were set is can mass spectrometry generate results similar to PCR? Can MS improve on costs? And can MS have a reasonably short turnaround? And that's 3 hours from sample arriving to data out at the end of it. And that's quite an unusual thing for a mass spectrometry based analysis. And not only that, can you do it in 3 months? And that perhaps was the real challenge in this. This brought together then a group of researchers in universities in what were called the pilot labs to develop the methodology. And this was to assess the methodology. This led through then to a phase 2 clinical pilot validation, which is going on at the moment in hospital labs. And if that's successful, then we're hoping that this will roll out in phase 3 to a UK-wide testing base. And this will be the first national rollout of a mass spectrometry-based testing program that there will have been in the UK. And the aim is to both cover population screening, so can we detect the antigen in diagnostic tests, but also does mass spectrometry offer anything else? So for instance, risk management, can we look at stratification of patients and their likelihood of developing term, and then long-term population health, can we see how things like long COVID are represented in the samples that we're getting from them? So you can see that we went from the original goal of population screening to some longer term goals than that. And this really is a huge collaborative effort. It could not have been done without the collaboration of all of these different people, the universities, the hospital labs, and the industries involved. And I hope you'll see that through the rest of these presentations. Originally for the Moonshot, these are the labs that were involved. You'll see these are all university labs involved in phase 1. So this is the development of the methodologies, and Dan's going to tell you a little bit about the work that went on in Leicester to develop some of the methodologies for analysing COVID. And then the ones in red here took this forward and have been continuing to work on the development of the methodology for diagnostics. But you can see already just how broad a range of different universities were involved, and a couple of those are Midlands universities, Leicester and Warwick, but it was much, much broader than that. These universities working as a coalition, and it could not have been done without all of those universities working together. The phase 2, the clinical labs are also absolutely critical to the process. Could we translate what was done in an academic lab in terms of an academically developed assay through to something that could be used in a standard clinical laboratory with the usual mass spectrometry equipment that's available in those laboratories and the expertise they have? And we have, fortunately, 10 hospital laboratories around the UK now testing out the methodology that was developed as part of the moonshot and looking for accreditation of that, those methodologies for taking forward into an accredited programme. I should particularly note on here the Guy's and St Thomas's in London, who've been absolutely at the forefront of leading this, and the Great Ormond Street team. They've been fundamental in taking forward this process, and then the University Hospitals of Leicester and Manchester have been very, very heavily involved as well. So that's an ongoing process. We don't have the outcomes of that yet, but what we're going to tell you about is what went on in the phase 1 part of this, of development of an assay and our understanding of what goes on in SARS-CoV-2. Just to give you an idea of how challenging this might be, when Dan starts to talk, we were asked to see whether we could pick up SARS-CoV-2 at a CT of 25. So you'll be familiar with these cycle number from the PCR. CT25 means about 80,000 genomes present in a 10 microliter sample, and if we assume that there are 300 copies of the NCAP protein, we're talking about identifying 40 attomoles of NCAP in the sample that we have available to us. So we really are talking about what's at the bottom end of what mass spectrometry, we thought mass spectrometry would be able to deliver in terms of its sensitivity. So just bear that in mind. This was the goal that we were working towards, is that low attomolar sensitivity for detecting SARS-CoV-2. So I'm going to stop there. That's my introduction. I'm going to now pass on to our main speaker today, who is Dan Lane from Leicester, University of Leicester, working in Don Jones's group down there. And Dan is going to take us through some of the incredible work that's been going on at Leicester in a very, very short period of time. If Dan looks tired, it's probably because he's been working 24 hours a day, 7 days a week for about as long as anyone can remember. And he's going to take you through some of the work that they've been doing down there. So I'm going to stop sharing my screen and pass over to Dan.
Nice one. Thank you, Andy. And thank you to the Midlands Mass Spec group. Really appreciate coming on here. Hopefully it won't be the last time I'm on here either, but we'll see how it goes. I'm just trying to share my screen.
Dan, while you're doing that, I'll just mention to people that if they have any questions along the way, there's a Q&A button at the bottom of the screen. If you could put your questions in the Q&A. We're going to leave it right to the end of the presentations to deal with the questions, so that you'll have seen Dan's presentation and the information I'm going to pass over. But there is a Q&A at the bottom. But keep an eye on that. Please do put your questions there. We have had some problems with that, so we may come back to you and ask you to put things in chat, or if you prefer to put things in the chat, just put them there, but try to use the Q&A if you can. Thank you.
Thanks, Andy. So just a brief intro on myself. My name's Dan. I'm a last year PhD student working at the University of Leicester through the Department of Health Sciences. Worked through Don Jones's proteomics group. My PhD was mainly based on metabolomics and also drug quantification. But then when the pandemic hit, I essentially put all that in the bin and then just swapped everything for all the COVID work that I'm about to speak to you about. So the presentation title is Developing and Implementing COVID Mass Spec Diagnostics for Clinical Use. So we're just going to speak about the journey that we've been over for the past year. So just a bit of background on the coronavirus structure. You know, a lot of you will already be quite familiar with this. There's two key components to it, really. There's the genetic material, so the single-stranded RNA that lives inside, and then the structural proteins, so nucleocapsid, spike, envelope, and membrane. There's also a couple of other non-structural proteins involved, but it's important to note what the structure of the virus is, because these are the targets for detection for us. So, you know, PCR is looking at the single-stranded RNA, whereas if we wanted to move something forward through proteomics, it would be for the proteins of the virus. So the NCAP protein makes up about 88% of the proteome, so that's a really good target, and that's the main one that I'll be speaking about today. So just a quick overview on what the initial detection system was during the beginning phases of the pandemic, and still now, to be fair, RT-PCR. So, you know, you take a swab sample, you put it in a universal transport media, that's transported to a lab. The RNA is then extracted, then amplified up, generating the cycle threshold value, which is detectable through qPCR. So initially, there are two parts of this process that could really be improved upon. So for mass testing, swabbing's not very useful, especially in care homes and schools, where it's quite hard to swab children and elderly people. So that's something we wanted to look into during the initial phases of the pandemic. And then there was a recent systematic review looking into the false negative rate of PCR assays, and ranges from 0.2 to 58%. But this is probably to do with the heterogeneity between the different tests. They're looking for different genetic sequences. People use different kits, different instruments, but still it's widely reported that the false negative rate could be improved upon. So it's something that we wanted to look into. So this is what we proposed, a schematic of what we initially wanted to do in May of last year. So, collect the sample by any means, saliva, using the residual swab fluid from the other samples, then triptychally digesting the protein, so NCAP or spike, down into peptides, and then analyze on a triple quad, and then seeing whether or not we could detect the protein. So initially in May of last year, we were working with Martin and Bart, who were from Ghent University. They were leading a Europe-wide mass spec community-based approach for COVID detection. Before we started working with them, they did a lot of in silico work to identify good target peptides from the NCAP and spike protein using components and proteomics, which is where we came in as the Bangis biomarker facility here in Leicester. So we were tasked with trying to help them optimize the assay that they'd make on our instrument. We're using the Xevo TQ-XS Waters, and they supplied us with a couple of recombinant proteins and peptides. So we use something called the MassLink Skyline interface, which is a clever piece of software that can essentially create a scheduled, optimized mass spec method on MLRM. I'll just talk a little bit about that. So using Skyline, we can load in the protein sequences and then export a mass spec file with predicted MRM transition. And then the MassLink Skyline interface then reads this, and then through successive injections of that triptych digest, it can create an optimized scheduled MRM method. So that was quite quick. That took us a day or so to do, although you can really do it on a multiplex sample in half an hour. So it's quite a useful piece of technology. We then looked into optimizing the chromatography. So we still use this 8-minute method that we started with occasionally because it's quite a good baseline for us. But we compared an 8-minute, 5-minute, 3-minute, 2-minute, and a 1.5-minute chromatography. And, looking through them, even down to the 3 and 2-minute, there's quite good separation of the peptides we were looking at. And these are the NCAP and spike peptides that we were interested in. And then to see whether or not that had any impacts on like peak area and detection, we found that generally steeper gradients were quite well tolerated in neat samples. So for a high throughput assay, which is what we wanted to create, we could even go down to 1.5 minutes. So we were quite happy with this. So then further on optimizing, last summer, we were looking into the digestion process and efficiency. We tested a 15-minute digest with no IA or DTT against a 15-minute standard with IA, DTT, a 1-hour standard and a 4-hour standard for both the NCAP and spike protein. Essentially what we found is that NCAP is readily digested in any circumstance. Even at 15 minutes without any IA or DTT step, we were able to get quite good digestion of the peptide. But with spike, I think because it's probably got a couple more cysteines on it, it takes a bit longer to thoroughly digest. But, as I said earlier, NCAP makes up about 88% of the proteome. So that's the target that we're really interested in. We also looked into different concentrations of trypsin, using the expensive trypsin lyse C, and also using, cofactors like calcium chloride. Top left graph, we can see for NCAP, these are different peptides and different total areas on the side. In any condition, they were pretty similar. So, lyse C made no difference to NCAP. Again, we proved that it was readily digested in any amount, even 0.1 micrograms per microliter. But because it also tolerated high concentrations of trypsin, and spike requires more trypsin, where it's recommended to use an excess. But essentially what we proved is that you could use a 15-minute digest to the proteins that we're interested in. So just to review, so this is where we were in midsummer last year. We'd created the scheduled MRM method using the MassLink Skyline interface. We'd made a 1.5-minute runtime on the instrument and a 15-minute digestion without IA or DT2. So, sample prep you could do within half an hour and then also run it within half an hour. So it's quite a high throughput method. So conditions on the mass spec, we're using reverse phase, C18 column, using water and acetonitrile, and 0.6 mil per minute flow rate. So then it was time to receive real samples. So we received a couple of residual swab fluid samples. So when swabs are taken, they're put into essentially what is a buffer, so as to keep the virus intact for the PCR lab. We were interested in looking in the buffer to see if we could see any residual protein. There were two main collection devices in use within Leicester last summer. They were the M4RT and the Viracult. So we were receiving samples from both of these. And we'd run 30 samples in total using the following method. Just taking 10 microliters of swab fluid, crashing with ice cold acetone, removing supernatant after a spin, and then doing the 15-minute digest that we spoke about before, then analyzing using the MRM method we'd made earlier. And essentially, just from the first attempt, we managed to find 2 positive samples. So I'll go through. So these are 5 peptides on the next couple of slides that were positive we decided were positive on the mass spec. So top left pane, we've got a spike standard. Top right, we've got negative sample, negative PCR sample. And the bottom 2 are the 2 patient samples that we detected. So this was the NCAP peptide LNQ. So highlighted in blue in the bottom 2 right panels, you can see a peak. Again, for the next peptide, KQQ, that's quite clear cut. DGI, another peptide, GFYA again, and so essentially we'd seen 5 peptides, all with multiple transitions. So we were quite happy that these 2 samples were positive on mass spec. But then looking into the CT values of these, the 2 samples we'd analysed that were positive. So Andy spoke a little bit earlier about cycle threshold, but essentially lower is higher, means higher load. And I'd probably say normally distributed range of a positive sample, probably from CT17 to CT30. And the 2 samples that we detected the virus protein in were 12.5 and 14.5 CT. So, these were really high viral loaded samples, but, essentially we proved the point. We're able to detect the protein in these samples, but it raised questions on sensitivity. I'll also say at this point, we also tested the sensitivity of our instrument, and we were getting down to about 50 attomoles on column. So I think there was a bit of an improvement to go with there. We think we were only able to see in the really high viral loaded samples because of the noise issues of these M4RT and Viracult buffers. Essentially, they're full of albumin and salt, which is a nightmare for mass spec proteomics. They had coelutin peaks and the noise is quite high. So in order for a viral peptide to break through, it would have to be in quite a high amount. Briefly, we had a look at the ionization suppression of those universal transport media, so the M4RT and Viracult samples. They both essentially enhance the signal of the peptides we're interested in. But at the same time, the noise is so high that it's a bit moot. So this is where we were late summer 2020. We'd created a high throughput mass spec method. We've successfully identified a couple of viral peptides from the swab fluids, although it was clear that we needed some sort of enrichment to increase the sensitivity. So then enter Operation Moonshot in late 2020. Andy's already introduced slightly. So essentially, just briefly, it's a DHSC directive there to create new diagnostics for the detection of COVID. Initially the intention was for mass screening, so high throughput assay. And what they had done, because most mass spec labs don't have access to high category fume cupboards, for handling live samples. We asked the DHSC to swap out the swab fluids for 70% ethanol so we could receive inactivated samples. And at the same time, this is removing quite a lot of the noise we were experiencing earlier. So we were quite happy with this. So yeah, so Andy's already shared this slide, but essentially there were 8 centres using mass spec to try and develop a collaborative method for the detection of the virus. I think 2 of which were looking at mouldy MS and the rest were looking at triple quads. But yeah, so yeah, we were one of the centres looking at triple quad because we'd already had the method. So the new objectives were to look into different matrices, so like saliva, swab tibs, and then also the new 70% ethanol swab fluid, and then explore cleanup strategies in order to improve the sensitivity. So just running the initial method that we'd created earlier in the summer, just to get a gauge of the lower limit of detection of the different matrices, we were detecting around 1 to 5 femtomole per microliter between the matrices we were looking into. So, you know, in human saliva, we could detect the NCAP peptides down to 5 femtomole per microliter. And as Andy shared in the slide earlier, the limit of detection really needs to be lowered at least 10 to 100-fold more to be able to start detecting those less viral intense samples. So, you know, the method was quite similar. It was just a suck and see, just to see where we were at, really. So running 80 microliters of sample, crushing with acetone, spinning down, running the quick digest, and then analysing. So essentially, we needed to enrich the sample to increase the sensitivity, and then came Ciscapa. So Ciscapa is a company and the product of the same name. Essentially, they create antibodies that are conjugated to magnetic beads. The antibodies are specific to NCAP peptides. So essentially, you get a sample, you digest your sample, you add in your magnetic beads with their antibodies, they bind to the digested peptides, and then you can wash off the noise and the supernatant of the sample and then run it on the mass spec. So, you know, it really allows for the separation of viral peptides from the dirty matrix, which is the issues that we were experiencing before. So yeah, just a brief one, just to show you what it really looks like in the lab. So we're running on 96 wall plates. So on the left, we've got the digest that we're incubating with the in solution magnetic beads. You then place that on a magnetic array after letting them bind for an hour. And then on the right-hand side, you can see the magnets pull the beads away from the middle of the well and allows you to get in with a pipette and remove the dirty matrix that we don't want. You can then obviously wash the beads and repeat the process to further clean. You then elute with formic acid, and then you can run straight on the mass spec. So as our role in Leicester as a P1 lab, we were mainly looking into saliva. So I'll just speak about the protocol that we were initially running with. So the total runtime was almost 3 hours, so sort of around the limit of what we wanted to do. A lot longer than the initial acetone crash that we were running, but you know, it allowed us to actually separate out these peptides from the background. So essentially crashing the saliva with the 70% inactivation solution, so just in ethanol, removing the supernatant, adding trypsin, digesting, then adding the beads, washing and eluting, then running that on the mass spec. So, you know, the selectivity that we got from Ciscapa was second to none. You know, here are the 3, with Ciscapa, we were given 3 sets of antibodies that bound to the peptides ADE, AYN, and DGI. And this MRM method that we're running, we're looking at 40 peptides. We found no co-eluting peaks. It removed essentially all the noise from the spiked human saliva sample. And it selectively pulled out AYN, DGI, and ADE. So we were pretty happy with this. Looking into the recovery, so we compared the recovery from human saliva to synthetic saliva. The important figure here is the one on the top left. So we compared pre and post spiked recombinant proteins after Ciscapa. Essentially what we found is that we lost about 30% of AYN and ADE after Ciscapa. But, you know, the trade-off of that selectivity, that was, you know, removing the interfering peaks of noise, was done without any substantial loss, so we were quite happy. So we tried this out in a couple of patient samples to start off with. So again, we've got standard top left that was spiked into a human sample, and then a PCR negative sample against 2 positive PCR samples. And I think this was the peptide DGI, and we were able to clearly detect samples using the SysGAPA protocol against PCR negative samples. So we were quite happy with it. So then through Operation Moonshot, they provided us samples from the Falcon study early January of this year. And we ran 319 saliva samples and tested them with our SysGAPA method. So there was 150 PCR positive samples out of this set. 63 of them we found positive on mass spec. So, you know, we'd come a long way in a year, and we've done sensitivity and specificity to track true false positives and true false negatives against PCR. And, you know, sensitivity was quite low in all the samples, so 34%, but specificity was quite high, so 93%. And, you know, the further, we broke this down into different CT values, so PCR cycle threshold. The lower you go, essentially, the higher sensitivity and the better the correlation between mass spec and PCR. But, you know, so there's still work to be done. The main issues with the test that we've got at the moment is the high sample heterogeneity in saliva. You know, the mucus content between each person is quite different. It varies from the time of day. And it's also quite hard to get someone to accurately spit out a mil of saliva into a tube. So we're getting highly heterogeneous samples, and the mucus content was causing us all sorts of problems. Quite hard with pipetting these samples, quite hard, like just general sample handling. And the mucus was also binding to the beads, preventing them from dispersing into solution. So that was also an issue. But, you know, a couple of the other P1 labs were mostly looking into swab tips, and they were using SysGAPA, greater sensitivity and specificity. I think above 80% and above 90%. I think Andy will probably clear that up later. But, you know, so essentially what we've done as part of the P1 phases, we've got a saliva and swab tip methods that could detect viral peptides in solution. The swab tip method was better, but we're still trying to develop the saliva test as we speak, and hopefully we can raise the sensitivity of it. And yeah, that's it. Thanks for your time, everyone. I think you're on mute, Andy. Technology,
eh? Yeah. So thank you, Dan. That's a really nice overview, and I think an overview of a complex journey broken down into a very small period of time. So I think, Dan, if you give me control, if back of the screen,
yeah.
What I'll do then is I'll follow on from where Dan got to and update you on where we are now with the project. One of the things that we wanted to do in these colloquia is to have a scientific talk, but also have a talk where we're honest about how good the methods were and some of the challenges that we faced along the way as we were developing the methods. And I think you've seen that from what Dan was showing you. So if I can get the slideshow to work. Okay, so I'm going to pick up from where Dan left off in a way. So this was the target that we were set by the NHS Test and Trace team to pick up 2 to 5 peptides from the samples to have an inject-to-inject time. So that's a sample analysis time on the mass spectrometer, including chromatography, of 5 minutes or less. And I think many of you will be taking deep breath when you see that as a challenge. That means that basically 3-minute gradient is about the most you can do. So for analyzing complex biological samples, is it really possible to do that in a 5-minute gradient? From the sample processing, they asked us if we could get it down to 2 hours, but at least 2 to 3 hours for the total processing from sample arriving in the lab to data coming out the far end. And as Dan has been through with you, you can see how challenging that was. So that meant that a minimum of 300 samples per day per machine, so that would be competitive with what we might expect from a clinical lab. And then, not asking too much, specificity of greater than 99% and a sensitivity of greater than 95% on samples with a CT value of around about 25 was what they were asking us to do. And I think you can see that that's quite a challenge for any mass spectrometry based analysis. The other considerations they asked us to think about as we were developing the method was scalability. Was it possible to supply the reagents at scale, which would be needed to analyze hundreds or thousands of samples a day? And was this robust over time? Could we actually do this over a longer period of time? And was the methodology robust? So that was quite a tall order when we all joined this coalition in September last year, especially because they wanted it done by Christmas. So just a couple of then follow on from what Dan was saying about the different ways that you can sample for SARS-CoV-2. We're all familiar with the oral nasopharyngeal swab. You can see the chap here having as much fun as I think all of us do, sticking a swab up your nose. Very uncomfortable process. This of course is very familiar to people because it's what's used for both the lateral flow and the PCR test. So people are becoming more familiar with this. Its advantages are it's taking a sample done directly from the site of probable highest load. That's from the back of the nasal cavity and from the tonsils. And there's minimal dilution of sample because you're sampling directly from that point. However, there are some cons with that. First of all, I mean, we all know how, I'm sure we've all done this, we all know how uncomfortable it is. So actually there's very variable compliance with swabbing. So how well do people actually stick the swab up their nose or to the back of their throat? And actually, I'll show you some data later that will give you an idea of just how difficult that can be. And it's a relatively small sample. All you have is the sample that's absorbed onto a small swab. So for a mass spec analysis of a complex sample, you're dealing with no more than 50 microliters of sample effectively absorbed onto the swab. So you're talking about a very small sample. So saliva sounded like a good alternative. Saliva has been used for many years for DNA testing and in testing for things like Cushing's disease. So it's well known as a biological medium to take samples from. The pro is it's very simple. You spit into a tube. The cons are, of course, that it's indirect. We're not necessarily sampling from the point of highest infection. There's quite a lot of sample dilution in the saliva. There's a variable concentration of everything in the saliva and the makeup of saliva, and you often get contamination of the saliva as well. So the picture here is just of 6 different tubes of saliva that we received. These have been precipitated in ethanol, and I think if you have a look at those, you can see the different samples that we got. Some of them were very well behaved, like the one on the left-hand side, but some were really badly behaved, like some of the ones in the middle. And I haven't included the ones that had lumps of carrot in them. So there was quite significant contamination. As Dan mentioned, one of the things that we also thought about was sample handling. And because we work in university labs, we were told that if we were handling live virus, which is what you get with the standard PCR test, we would need to work in a category 3 lab. And actually most universities don't have that. So we decided to inactivate at the point of sampling. So you can see all of these samples here are actually in 70% ethanol, so they've all been fully inactivated. And this has some big advantages in terms of the supply chain and getting samples across. But you can see that even at the point of what samples do we use, it's not simple to decide which samples to use. And in the end, we ended up trying both of these to see which was the best. So what were the unexpected challenges? Well, I think certainly in Manchester, where I'm based now, the biggest challenge was becoming a logistics hub. What we didn't realise is not only would we have to do the analysis, but we'd also have to make up all the sampling packs, send them out, get them back in and distribute them around the laboratories being tested. So this is a picture of the foyer at the MIB, Manchester Institute of Biotechnology, and a group of people converting boxes of plastic bags and tubes into 15,000 sampling packs that we then could distribute to get the sampling done. So it wasn't that somebody else did this for us. Actually, we had to generate the sampling packs ourselves. Part of the challenge with that was also changing materials due to supply chain. So the swab that we were using in the middle of the program changed from a small swab to a larger swab and then back to a smaller swab just because of what was available in the supply chain. And of course, that changed the amount of material that was coming through into the assay. So, so even supply chain had an effect on what we were doing. Another challenge we had was the success of the lockdown before Christmas. If you all remember, perhaps if you think back, we had a lockdown and it was very successful in suppressing virus. And actually at the point when we were ready to go, the number of cases presenting at hospital started to drop very, very significantly. So it actually became quite challenging to get positive samples. And so for the first month and a half of running the program, we were unable to get large numbers of positive samples. And well, when we set up the program, there were many around and we thought this wouldn't be a problem. It turned out to be a problem. And it's something that you can't necessarily predict is in a pandemic of this sort is sample supply. One of the things that we've also learned is if someone's going to compare, if someone says they're going to compare your results to a gold standard, take a step back and ask them what that gold standard is. So in this case, the gold standard is the PCR test. And the plot at the bottom right-hand side of the slide here is one that we haven't published yet. But this is data where swabs were taken from patients and analysed by PCR in 2 different centres. I won't mention what the centres are, but the orange dots are one centre and the blue dots are the other centre. Across the bottom axis is CT value, and on the vertical axis is the difference in CT value measured in the 2 different centres for samples from the same patient. And so you can see that some of these are 8, 10, 12-fold difference in CT value, which means one CT value is a 2-fold change. So we're talking about 1,000 to 2,000-fold difference in the amount of virus being measured in the same patient on the same day taken by swab. So whether the PCR test is a gold standard or not, I think is open for debate. And of course, that meant that we were being tested against something which itself wasn't an incredibly reliable test. Just so you know, the yellow dots in the middle of that plot are actually the comparisons of the values that have come through from the mass spec assays. And you can see how much more tightly clustered those are. And those are taken from the 2 or 3 labs across the UK that ran the same samples at the same time. And you can see that actually mass spec did a much better job in terms of being close together. I've already mentioned the very tight timelines we were working to. Obviously, Operation Moonshot, the aim was to deliver testing very, very quickly so that we could get the population back out of lockdown. And those timelines became even tighter as we had challenges with sample supply. And then conflicting views on how we should translate and what you were able to do in a clinical biochemistry lab. So one of the things we discovered is that when you work with clinical biochemistry labs in the hospitals, they have different experiences. And some of them will say, yes, we can do that, no problems. And others will say, what's that? So you have to bear in mind the different experiences that people will have when you're going to translate this through into a clinical laboratory. Anyway, as Dan has mentioned, we came up with this harmonized method. This is the method that effectively Dan has described to you, so I'm not going to go through it in detail. The key to this was the cis-kappa enrichment, which allowed us to get down to that low attomolar concentration that we needed to in order to get the assay to work. So where did we get to? Well, these are the consortium results for the swab. So about 550 samples were run and batched into 96 samples. And you can see there are 2 tables here. This is results from 2 different labs. And the key thing is the numbers in red. These are the sensitivity and specificity that we were able to get from the swabs. And you can see that 97% sensitivity and 96% specificity are very, very close to what we needed to be in order to be competitive with what was called the gold standard. In fact, if you do the sums, this is better now than the gold standard. So certainly the outcomes of that development program were that the swab is giving data which is at least as good as the gold standard PCR data, but it's doing that in a short period of time. Results for saliva have been less good. I'll say that straight away, but you've seen these numbers. These are the numbers from Dan, and you can see that we're getting pretty close to where we need to be in terms of sensitivity and specificity. It's still quite low in terms of sensitivity, but we are still working on this, and those numbers are creeping up all the time as we improve the protocols, and especially we now have new reagents from Ciscapa, which look like they may make a significant difference. So some of the key metrics. We are close to getting some of this method accredited, so UCAS ISO accredited in the clinical biochemistry labs, and that means that it can then be used as a national test. We are working with the deactivated samples, so we do not need to work in class 3 hoods. We can work in class 2 laboratories, and probably even lower than that once we've actually developed this methodology. And that's makes a huge difference, because now those samples can come in and be handled without any really significantly special precautions. The cost, well, we're running around about £10 per sample. And if you remember, we were talking about £50 to £80 per sample for PCR. So actually, it turns out that even though there's an initial investment in mass spectrometry up front, by the time you take into account how much cheaper it is per test, mass spectrometry for mass testing has some great advantages over PCR. We now have a capacity of probably 500 tests per machine per day. We've also done some tests on pooling samples, because as the number of positive tests drops, we can start to pool samples, and we can probably get up to around about 10,000 samples per day with pooling. So mass spectrometry really can deliver that mass testing that was part of the key for the Moonshot project. And what skills are required? Well, most of the clinical labs are using a band 5 lab tech to do this work now. So actually, it really doesn't require high technical skill in order to get these sorts of numbers. So did we get to where we wanted to be? Yes, despite the challenges, everything that's green on here, we managed to achieve. So you can see that all of it was achieved, apart from the specificity, where we're very slightly off the 99% that was asked for us for the swab. But actually, we think a lot of that is down to the two actually incorrect PCR values that we're getting. Saliva is getting there, but we're not there yet. We completed phase 1 in 16 weeks, so twice as long as we were given initially, but that was due to that problem with sample availability. And phase 2, the rollout into the clinical biochemistry labs, will finish next week, and we'll hopefully then transfer that to accreditation and then to a national rollout. So yes, we can actually develop proper diagnostic tests for mass spectrometry in a short period of time and deliver that as a mass testing technology. But there were some other questions that were asked, which are on this slide. Can MS detect variants of concern, for instance? Can MS multiplex? So can we do things, can we not just look for SARS-CoV-2, but can we look for some of the other confounding diseases, such as flu and some of the other winter respiratory virus panel, which gives similar symptoms to COVID-2, but are not SARS? And mass spec has the opportunity to do that multiplexing, which in a single sample. And can we actually use mass spec to say, did the person swab themselves properly? Did they actually really engage that swab with the back of their nose and their tonsils, or did they just sort of stick it in their mouth and wave it around in the fresh air a bit, which is what I tend to do if I'm feeling a bit nauseous? So these are questions that we've also been addressing as part of the consortium. And so we've shown that the MS assay has some prognostic value. So we can actually, the amount, because we are using an internal standard to quantify the amount of material we have present, we can say that the amount of virus that we're measuring actually does correlate to patient outcome. Pooling works well, and we can pool 1 in 10 for swab and 1 in 5 for saliva and still get the same results. Dan talked about the Ciscapa enrichment, which pulls out those peptides that we are using to target SARS-CoV-2. But of course, once you've done that, you still have all of the other peptides left in the supernatant from that affinity enrichment. And we've been analysing those supernatants to see what else we can see. And we have now detected in those one of the variants of concern. This is the Kent variant, where there is a mutation of the glycine in this sequence here to an arginine which is then cleaved after. So we lose this GT-SPAR peptide and we pick up a T-SPAR peptide in, so TSPAR is the sequence of the peptide. We pick that up, and we have now seen that in a number of samples. So we think we can pick up the variants of concern. And we also think we can pick up flu. This data on the right-hand side here is a couple of samples which showed significantly high values for neuraminidase from influenza. So we think we can also multiplex the assays. So not only can we do what we were asked to do when we set out, but also mass spectrometry, because of its ability to multiplex and the fact that once we've generated data or samples, we can use that data or those samples to be reinterrogated. We can actually answer a lot more questions than some of the standard tests that are available today. So I hope that through that, I've convinced you that mass spectrometry has a real place in diagnostic testing and in diagnostic testing for pandemic outbreak or even for standard winter virus panels. I'm not going to go through this slide, but there are more things that we can think about once we realize mass spec can be used that we can go on and do. One of the things I should do is thank everybody who's been involved. The key to the success of this was working in partnership with instrument manufacturers, and I should mention Waters, Agilent and SciEx particularly, and especially Waters, who invested a huge amount of money and effort in the initial, especially the relationship with SysCapa and the development of those assays. The standards laboratories, the NML and LGC, have been absolutely critical in making sure that we were able to reach the standards that were necessary for UK accreditation. The team at SysCapa have been fantastic in developing these affinity enrichment methods, which allowed us to get to the levels that we needed to be able to be competitive with PCR. And then the driver behind this, the NHS Test and Trace, the DHSC, who are the funders of this, without whom it could not have been done. I could say thank you to the people. This is just some people of some pictures of some of the people who've been involved and some names. I started making this slide. I think I got to over 100 names of people who've been involved in this process in order to get us to the point that we are now in such a short period of time. So it does require a huge amount of investment in order to do this. And before the COVID outbreak, government would never have done this. They would never have put together this team in such a short period of time and funded it in a way that would actually be able to deliver it. It's only because of the SARS-CoV-2 that we've had the opportunity to demonstrate just how powerful mass spectrometry can be. So one of the real positives that may come out of this is the use of mass spectrometry in clinical laboratory diagnostics may now become much better established and much more broadly established than it has been in the past. And we now have a UK-wide team set up, ready to go, should we need to develop methodologies more broadly than we have now. So that's where I wanted to stop for today. So that hopefully has given you a really good overview of the project. There are a few questions in the question and answer, so I'm just going to have a quick look at those. The first of those is did we use any internal standards? As I mentioned, yes, we did. We used heavy internal standards for all of the peptides, so we were doing basically absolute quantification. There's a question from Corin Spickett. Were the ADE, DGI and AY peptides pulled out by SysCapa as the same ones we developed the MRM2? Dan, do you want to answer that one?
Yeah, so yeah, exactly the same. I mean, initially we dropped DGI from the panel that we were looking at because we weren't seeing it in any real samples, but SysCapa had created antibodies to AD, DGI and A1, and then we were able to detect all three of them in real samples. So yeah, it's the same one.
And I should add to that those peptides were chosen because they were when we did the initial testing, they were very intense, and the Ghent group and broader groups had seen them as well. Working with SysCapa now, we have monoclonal antibodies to I think five or six peptides now from the nucleic acid protein from SARS, and we're just testing those at the moment. So we're going to a slightly broader panel. And the reason for that is that we found with the very, very fast chromatography that we're using, there is some interference. There is a potential for interference, especially from saliva with the samples. So we're looking at some alternatives to that. And there's another question from Corin. Would it be good to get people to gargle rather than just spit? So this is where the collaboration with the clinical labs becomes really important. And not just with the clinical diagnostics labs, but actually with the clinicians who were collecting samples for us across the UK. Because we asked this question, and this was something that was discussed. The problem with gargling, especially with something that's as infectious as SARS-CoV-2, is when you gargle, you generate an aerosol. And so they were the hospitals were extremely reluctant to use a gargle as a way of collecting samples, just because of the potential for generating that aerosol and because of the problems they've had with infectivity of the SARS-CoV-2. So yes, that would be an ideal way of doing it, but at the point of sampling, we got a very clear message from the hospitals that no, they didn't want us to go down that route. So that's where one of the real strengths of this program has been, is that very, very close contact with everybody in the chain, from sample collection right the way through to the clinical diagnostics lab, to make sure that what we were doing would be fit for purpose once it was translated through to the hospital. Okay. Was mouldy TOF considered as a high throughput MS technique? I think, as Dan mentioned, sorry Dan, I'll take this because I'm not sure. Yeah. So I think, as Dan mentioned, if you saw the slide on the labs around the UK, two of the labs, Reading and Warwick, I think, were looking at mouldy as a potential route for this, because obviously mouldy in theory could be much higher throughput, and we had less success with mouldy than we had with the LC separation that we could get with electrospray. So the interference in the mouldy was much higher than we were getting with the electrospray. Now that was in the early phase of the project, when we were using polyclonal antibodies from Ciscapa and we were still developing the methodology. So it probably is worth us going back and having another look at mouldy as a potential route. Now we have the protocols worked out and the enrichment methods. But as it happened, we tried quite hard and it didn't really deliver for us. But I'm not saying it's not possible to develop a route, but we had to make a decision. We decided to go down the route of chromatography and ESI because that gave us the most promising results initially. Okay, well, I see we are up to time now, and there are no more questions coming in. So I don't know whether I should pass back over to Alex, or...
I just wanted to say, I will pass back to you to sign off, Andy, but I just wanted to say that this is obviously an ongoing seminar, and we are looking for speakers and also subject areas. So it doesn't have to be COVID related, anything to do with mass spectrometry. We are interested in running seminars on. So if you can contact either myself or Andy, I think both our emails are on the original invite that you've had. If not, I'll pop it in the chat. My email, and yes, please do get in touch, and we will look forward to the next one.
Great, thanks for that, Alex. And I would say that you know, this is the first in what we hope will be a series of seminars. We thought COVID was still current, and because of the success of the universities within the Midlands for doing this, then we thought we'd talk about this today. But really, the Midlands Mass Spectrometry Group is interested in mass spectrometry of any sort, applied to any problem, and whether, you know, whether that's petrochemicals, drug discovery, clinical, whatever. So we really would like to hear from anybody who would like to present their work. What we're going to try and do in these colloquia is a little bit what we've done today, which is to have a brief introduction, to have a scientific presentation like the one Dan gave you, but then also to have a bit of a technical discussion at the end of the meeting to see if we can, you know, to tell some of the truths that lie behind the methodology development. I think there's one more question is actually, okay, no, that's just as thanks. Okay. Chat is disabled.
I made it live just in case anybody did want to post anything. Bit late.
Okay. We live and learn every time we do this, I think. Anyway, so I think without more ado, I'd like to say thank you to Dan for really fantastic talk today. It is what he summarised there has given him many sleepless nights over the past few months, and belies the effort that went on behind the scenes to get us to where we are today. But I think he gave a really excellent overview of how you can take what was a very crude assay to start with and polish it to the point where it becomes viable as a clinical assay. I'd like to say thanks to Midlands Innovation again for hosting us today. They've done a lot of work behind the scenes, especially Alex, who's been involved in helping us set up the Midlands Mass Spectrometry Group and in keeping us on the straight and narrow. I'd like to say thanks to all the Midlands Mass Spectrometry Group members who've been helping out and keeping the Mass Spectrometry Group going. And last of all, I'd like to say thank you to all of you who've come along today and listened. I hope that was useful to you and you've learned something. If you do have any questions about today's talk and you wanted to do it offline, you've got our email addresses. If you want to talk to me about anything to do with Midlands Mass Spectrometry, please do get in touch and I'd be very happy to talk to you. So thank you all for attending.